Ayurvedic metal nanoparticles could be novel antiviral agents against SARS-CoV-2. This is printed in link.springer.com. Printed initially in international nano letters. Purpose of review. The pandemic COVID-19 has affected more than 70 million people globally. The whole world is eagerly waiting for effective antiviral therapy to combat COVID-19, but is yet to get it. The emergence of COVID-19 makes imperative the need for safe and potent antiviral drugs. Many metal nanoparticles exhibit significant antiviral potential against many viral diseases. The Ayurvedic system of medicine is the treasure of many metal nanoparticulate drugs termed as plasma. We'll look at the possible mechanism or action of the metal nanoparticles against coronavirus. It's a disruption of the outer layers of the coronavirus. Swana Basma, Rajata Basma, Tantra Basma, and Yashata Basma are recommended for COVID-19 treatment due to the ability to reduce the plasma, interleukins, interferons, and TNFs levels. Summary. The Ayurvedic Basma preparations are unique metal nanoparticles. These metal nanoparticles are safe, stable in solid state, and are having excellent biological activities. Ayurvedic metal nanoparticles, Swarna Basma, Rajata Basma, Tamra Basma, and Yashata Basma could be proven as novel antiviral agents against SARS-CoV-2. For their anti-inflammatory, immunomodulatory, antiviral, and adjuvant activities. Introduction. The search for effective treatment against COVID-19 is still wide open, though many different antiviral agents are tested or under testing in clinical trials to assess their efficacy against the SARS-CoV-2. In this context, Ayurveda, the traditional Indian system of medicine, may play an important role. This system has the potential to combat COVID-19, and the needs are conducting research activities, proper monitoring, and data keeping during implementation. In recent times, virus infections are posing significant global health challenges. Several epidemics occurred in the past few years due to viruses. MERS, SARS, swine flu are a few of those. And so many antiviral therapies and vaccines are available. Yet it's considered the development of a safe, effective, and potent antiviral therapy is in need of time. The reasons are being the occurrence of resistant virus strains, the adverse effect of the present therapies, and the weak response of the vaccines. There is a need for an effective antiviral medicine urgently. Nanotechnology may play a vital role in the prevention and treatment of COVID-19, and the metal nanoparticles have enormous potential to combat COVID-19. The emergence of the COVID-19 as a pandemic again makes imperative the need for such safe and potent antiviral drugs. The metal nanoparticles exhibit significant antiviral potential against many viral diseases. Metal nanoparticles can block cell attachment and viral entry into cells. It can inhibit viral proliferation and replication, and can cause viral inactivation and having virucidal effects. Many metal nanoparticles are used in therapeutic entities in Ayurveda for hundreds of years. In this context, the Ayurvedic Basma preparations, which are biologically produced metals and mineral nanoparticles, have the potential to emerge as novel antiviral agents against SARS-CoV-2 by their unique chemical and physical properties. The probable role of the plasma preparation for combating COVID-19 is searched and discussed in this review article. Metal nanoparticles are studied for the past few years for their antiviral activities, and many metal nanoparticles are found to have significant effects on many viruses. These nanoparticles are very effective antioxidants and can influence the production of ROS species by acting as ion reservoirs for the controlled release of bioactive ions. They are capable to go through the cell membrane due to very small size and can inhibit post-attachment virus replication. Gold nanoparticles can bind with the envelope glycoprotein of HIV and inhibit virus replication. It also shows antiviral effects against subtypes of influenza virus, H1N1, H3N2, H5N1. Gold nanoparticles were evaluated as a vaccine platform against the West Nile virus to assess the immune response. The gold nanoparticles were coated with West Nile virus envelope protein. The antivirus antibody was measured after inoculation in mice. The silver nanoparticles are reported to have significant antiviral activities against a wide range of virus like human immunodeficiency virus, hepatitis B virus, herpes simplex virus, monkeypox virus, and respiratory syncytial virus. The silver nanoparticles exhibit a virucidal effect in inhibiting virus reproduction. It can interact with cell receptors and prevents virus entry into the host cell in case of H1V1, HIV-1 virus. It can also inhibit double-strain RNA virus replication by interacting with the virus genome. The silver nanoparticles at a concentrate of 3.125 to 12.5 reduce the virus titer and the number of apoptotic cells elicited by transmissible gastroenteritis virus. Copper compounds, nanoparticles in oxide, iodide, and sulfur forms exhibited significant antiviral activities against herpes simplex virus, H1N1 influenza virus, and human norovirus, respectively. The copper oxide nanoparticles can reduce the viral population. Copper iodide nanoparticles inactivate activity of H1N1 influenza virus. Copper sulfide nanoparticles show a virucidal effect against human norovirus by degrading the capsid protein. The silver copper alloy nanoparticles can also inhibit HIV replication by binding with envelope glycoprotein GP120. Metal oxide nanoparticles such as iron oxide, zinc oxide have demonstrated specific antiviral activities. Iron oxide nanoparticles exhibit antiviral efficacy against human viruses. Metals form components of several viral proteins and play an important role in the survival and pathogenesis of the viruses. The nucleocapsid protein transactivation response, TAR, RNA interactions in viruses, metal ions are needed. Magnesium, zinc, and copper are the commonest metal ions that bind with the viral proteins. A recent study reported that a gold drug, auranofin, reduced the expression of the SARS-CoV-2 induced cytokines in human cells. It is an FDA-approved drug that contains gold with triethylphosphine, used for the treatment of rheumatoid arthritis. The study claimed that it could be a useful drug to limit SARS-CoV-2 infection and associated lung injury. A study reported that zinc combined with pyrethroid can inhibit the replication of SARS-CoV-2 by impairment of replication of the RNA viruses in cell culture. Zinc exhibited activity against RNA polymerase of multiprotein replication and transcription complexes of SARS-CoV and can have significantly inhibit the replication. A hypothesis is made that a combination of chloroquine or hydrochloroquine with zinc and an antibiotic like azithromycin could be an effective remedy. The reasons behind this combination are the broad availability, affordability, and demonstrated the efficacy and safety of the combination. Metal nanoparticles affect SARS-CoV-2 targets. The possible targets are very important for the development of a novel drug to combat SARS-CoV-2. Different studies have identified various structural targets of SARS-CoV-2, and most of the studies targeted spike protein. The other targets include RNA-dependent RNA polymerase, main protease, and non-structural protein. The target protein structures of SARS-CoV-2 are essential for drug designing against it. The same complexes of gold nanoparticles with TGEV antigen, enhanced respiratory activity of the peritoneal macrophages, and higher plasma IFN levels in comparison to the attenuated virus alone. Vaccination with the protein antigen and gold nanoparticles in an avian model of coronavirus infection increased the splenic T cell response, produced stronger antibodies, and enhanced the lymphatic antigen delivery in comparison to vaccination with inactivated virus vaccine. Gold nanoparticles were studied as vaccine adjuvant against SARS-CoV coronavirus. The protein of the coronavirus was targeted for blocking coronavirus infection. The gold nanoparticles were used as the carrier of the antigen and as an adjuvant of the subunit vaccines. It was observed that the antigen adjuvanted by gold nanoparticles exhibited strong antigen-specific IgG response against SARS-CoV infection. The Ayurvedic system of medicine is the treasure of metallic and mineral nanomedicines. A branch of Ayurveda, Rasa Shastra, deals with preparation and therapeutic application of nanomedicines, especially metallic and mineral nanoparticles. These metals and minerals are processed with various organic materials and repeated heating, and are finally prepared. Metal or mineral ashes are claimed to be organically structured nanoparticles. This prepared organometallic or organo-mineral nanoparticles are termed as bhasma in Ayurveda. These preparations are reported to be safe for human use and have various biological activities. Swana bhasma, incinerated gold, analyzed by X-ray diffraction and transmission electron microscope, reveals that the size of the crystallites within the Swana bhasma is approximately 30 to 60 nanometers. Rajata bhasma, incinerated silver, by scanning electron microscopy, analysis shows the regular and uniform arrangement of a cluster of granules in Rajata bhasma. The sizes of the particles range from 10 to 60 nanometers. The X-ray, Naga bhasma, incinerated lead, contained nanoscale structures on the surface. The width of the nanostructures lies between 50 and 60 nanometers, while the length ranges from 100 to 160 nanometers. The SEM images suggest that Vanga-Bosma incinerated tin particles show granular appearance and porous morphology. These nanoparticles have spherical morphology with the size smaller than 100 nanometers. Particle size analysis by XRD shows that the mean particle size of Vishada-Bosma incinerated zinc is 49 to 80 nanometers. The particles are smooth, spherical, and present in aggregates, as suggested by SEM analysis. Ayurvedic Bosma for COVID-19 treatment. Several Ayurvedic Bosma preparations are mentioned to be useful for COVID-19 treatment. These preparations are effective through sublingual and oral route due to better bioavailability and absorption on account of the presence of nanoparticles. Siddha Makarwaja nanoparticles and Swana Bosma nanogold particles are mentioned to be effective through administration with black pepper powder and ghee for treatment of moderate to severe COVID-19 patients. Many Ayurvedic formulations are proposed for preventative and curative purposes to combat COVID-19. These preparations contain Swana Bosma, gold nanoparticles, Rajata Bosma, silver nanoparticles, Tamara Bosma, copper nanoparticles, Lao Bosma, iron nanoparticles, and Vanga Bosma, tin nanoparticles. Immunomodulatory therapy. Drugs having immunomodulation effects are proposed for COVID-19 therapy. Many Ayurvedic Bosma preparations are having immunomodulation effect. Swana Bosma exhibits immunostimulant activity on macrophage function and promotes phagocytic activity. The experimental evidence indicates that Swana Bosma can be useful to improve the immune response. Rashada Bosma exhibits T-cell mediated immune response modulation. The immunomodulatory drugs may downregulate the adhesion molecules of the cell surface and reduce pro-inflammatory cytokine production. These drugs may stimulate phagocytosis caused by alveolar macrophages and inhibit neutrophil activation and mobilization. With the cytokine storms causing injury and exasperation is common in COVID-19 cases. Therefore, drug agents which inhibit inflammation are effective against COVID-19. Many Bosma preparations have inflammation inhibition activity. Swana Bosma, Rajata Bosma, Tamra Bosma, Rashada Bosma have inflammation inhibition activity. Swana Bosma is an anti-inflammatory drug that reduces cytokines production and stimulates both cellular and humoral immune response. It can influence T-cell activity, thus increases vascular permeability and encourages vasodilation. Tamra Bosma is reported to have significant inflammation inhibition activity. This is achieved by reduced production of interleukins and TNF. Antiviral effects. Some metal nanoparticles exhibit a virucidal effect by destroying the envelope of the viruses or by inhibiting virus reproduction. Swana Bosma may affect the spike protein and can disrupt outer layers of the coronavirus. Rajata Bosma can inhibit reproduction of novel coronavirus. Tamra Bosma may exhibit a virucidal effect against SARS-CoV-2 by degrading the capsid protein. Vaccine adjuvant. The Ayurvedic Bosma preparations have Rasayana immunomodulatory effect, may be used as vaccine adjuvant. Ayurvedic immunomodulators can be administered along with COVID-19 vaccines to elicit a faster and stronger immune response. Swana Bosma containing gold nanoparticles may act as an effective vaccine adjuvant for the stronger immune response with the COVID-19 vaccines by increasing levels of IFN, IL-1, and IL-6. The other Basma preparations with the immunomodulatory effect, like Rishada Basma, may also be effective as vaccine adjuvant to increase immune response. A hypothesis has been drawn that silver, copper, and zinc nanoparticles will also be effective to fight against COVID-19 due to their antimicrobial properties to prevent contamination and contagion. Emphasis has been given to the use of copper salt nanoparticles for having antiviral effects. By considering this hypothesis, it may be inferred that Rajata Basma, Tamra Basma, Rishada Basma all have the ability to reduce the plasma interleukins, interferons, and TNF levels. The efficacy of these nanomedicines can be evaluated by determining these plasma biomarkers before and after treatment. Conclusion: research works on uses of metal nanoparticles for combating COVID-19 are merely sporadic, although the metal nanoparticles have enormous potential to affect the possible therapeutic targets of SARS-CoV-2. Ayurvedic metal nanoparticles, especially Swana Basma, Rajata Basma, Tamra Basma, and Rishada Basma, could be proved as novel antiviral agents against SARS-CoV-2 due to their anti-inflammatory, anti-immunomodulatory, antiviral, and adjuvant activities.